Welcome, everyone. Once again, thank you for joining me today as we go through the next webinar that I have put together. I hope everyone's doing well, staying healthy, practicing good personal hygiene, self-care, physical distancing, all the things that we've been asked to do. This is still important. I've just heard that here in Massachusetts, we're being asked to continued to stay at home past the originally stipulated time frame, which was due to expire next week, but for another 2 weeks. So this is still very much our reality here in this state. So um, carry on, and we will carry on here, Pioneer Valley Osteopathy, with this next offering. So I thought it would be a really neat exploration to go through some of these functional medicine tests that I make regular use of in my practice um, as a way to gain insight that's not typically available through conventional um, medical testing sort of channels. Uh, one of my favorite tests by far would be the Comprehensive Stool Analysis. And sort of superimposed on this idea of wishing to go through some of these tests is wanting to just bring into your awareness some ideas for tests that can be done at home right now when we are being asked to stay at home for what looks like an additional 2 weeks at this point. So again, this test, again, it's one of my favorites, is done at home and is really amazing, as I hope you will agree by the end of this presentation. First and foremost, I just want to state right off the bat that I have no beneficial financial relationship with any laboratory testing company referenced in this presentation. Um, the fees for these tests are handled exclusively between my patients and the lab companies. The labs themselves are doing great work. They deserve to be supported. And if it makes sense medically, I want to do what I can to facilitate my patients' desire to gain the insights available through this type of specialty testing route. Secondarily, I just wanted to provide a very brief update at the top on where things are standing with respect to COVID-19. We've been doing this with each webinar each week. The latest count of confirmed cases worldwide is a little over 3 million. You can see how that's grown. We didn't really do this last week during the webinar because it was a shared platform, but from 2 weeks ago, it was just under 2 million. The fatality rate is continuing to go up as well. But again, as we talked about last week, that has to do with confirmed cases as opposed to, what the research is really starting to bear out, the likelihood of there being far more cases than have been legitimately confirmed by scientific studies. But we spoke about this about 2 weeks ago. This one study that came out at the start of April really seems to think that cases may be underestimated by 2 to 3 orders of magnitude, which is really quite something. And then just last week in the New York Times, there was this article, which was based on some studies of serologic testing that were undertaken throughout the whole state of New York. 3,000 people looking for antibodies to this virus, the SARS-CoV-2 virus. Statewide, the prevalence came back at almost 14%, and in New York City, above 20%. So 1 in 5. And if we extrapolate from that, we see that the number of cases inferred from this study, just in the state of New York, 2.7 million. Compare that with the confirmed cases worldwide at just over 3 million currently. The key takeaway, apart from the significantly reduced mortality rate that we can infer from these numbers, is that we just don't know yet at this point whether having been exposed to this virus actually yields a functional immunity to subsequent exposure to the virus. We need more research. We need more and better understanding about this virus and how it works to be able to say whether or not that will hold true as it has with respect to so many other infectious diseases that we've encountered and that we are vaccinated against. So to be determined. There was another study in New York that was looking at some hospitalized patients. This is important because I think it really highlights this key detail that I've been speaking about during the first two of these webinars, basically pre-existing conditions and what that says about increased risk factors. So 88% of these 5,700 hospitalized people had 2 or more pre-existing conditions, while fully half of them had 4 or more pre-existing conditions. Most of these people hadn't smoked. Many of them had asthma, not a huge percentage. Some of them had COPD as well. But you can see that the bulk of these pre-existing conditions were listed there at the bottom of the graph. And the most common ones were high blood pressure, diabetes, and obesity. Now, why is that particularly relevant? Well, as was stated by the physician on whose website I read about this study, the virus doesn't seem to be targeting patients with pre-existing lung problems so much as those with a pre-existing tendency towards inflammation. All of these most common pre-existing conditions, high blood pressure, diabetes, obesity, are significantly characterized by the presence of inflammation. And that's what a lot of those preventative measures that I suggested a couple of weeks ago really are seeking to address, reducing overall systemic inflammation. As Dr. Herskowitz said, the best way to prepare for the future, any future, is to focus on us, the hosts, not the virus. Because again, we just don't know if, at least at this point, if having been exposed to the virus makes it so that you can't come down with COVID-19, the actual complex of very potentially serious repercussions of exposure. So now, the reason why we are here, let's talk about the gut, or as it would be referred to in more highfalutin medical speak, the alimentary canal. I think we're all broadly familiar with the topography of the gut, more or less. I wanted to just start this webinar like any good medical lecture would start with a little bit of anatomic review. This is just sort of here in the bottom left, a schematic of a prototypical segment of the gut. As I often say to many of my patients, even though it's inside of the body, it is still technically outside of the body. It's the outside world. It's not subject to the high degree of security and control that, say, our organs, tissues, and bloodstream is. So it's, yes, inside of our torso, but it's still technically the outside world. On the right side of the screen, you can see this schematic of these finger-like projections that characterize the small intestine in particular. We call these villi, but basically they are a way of increasing the available surface area of the intestinal mucosa, that interior lining of the gut, which is so important for absorbing the nutrients that we take in the foods that we eat. This availability of surface area is actually significantly increased by the presence of these villi, one of which is sort of depicted here, but also by these tiny little projections that extend from each one of these cells. You can see the cells roughly approximated here in this schematic drawing as individual little units. And then from the end of each one of these are what we would call microvilli that further increase the surface area. Now, the small intestine itself is only about 20 feet long, the large intestine about 5 feet long. But the overall surface area that is produced over this approximate 25-foot length of tubing is about the size of a tennis court. And that's pretty impressive, to my mind at least. It is indeed the largest mucosal surface area in the body. And because it is such an important interface between the outside world, again, inside the gut tube is the outside world, and our protected inner space, the body, in its inherent wisdom, has chosen to put 70% of the immune system just on the other side, and in certain cases, starting to extend through, which you can see a little bit here in this drawing. Because that's that's where the outside world meets our protected inner space. And we want all of our first responders, so to speak, ready to go at the initial signs of any difficulties. Now, we've probably, all of us have heard this term microbiome. What does that mean exactly? It gets used a little bit inaccurately. Technically, it refers to the sum total of the genetic material of all the microbes in and on their human host. This can refer to genetic material from bacteria, archaea, which are similar to bacteria, but definitely different, fungi, protozoans, which are also known as parasites, and viruses. There's approximately 200 times the number of genes in this overall microbiome pool as there are in the human genome. That's pretty impressive. And there's maybe, the best guess is somewhere between 3 and 10 times the number of actual individual cells of these symbionts as there are actual human cells in the body. Microbiota is the term that's used to represent the individuals within these communities of microorganisms. That's, I think, how most people are intending to mean the usage of the word microbiome when they use it, but scientifically, technically, the term is microbiota. Now, what do these gut bacteria do for us, their host? Well, quite a few important functions in actuality. Some of the more important ones are that they actually serve to outcompete some very potentially bad acting bacteria. They help to modulate, train up, calm down the immune system, which again, 70% of which is just on the other side of that one cell layer thick barrier that separates the outside world from our protected inner space. And they're also very important in helping to digest, absorb, produce certain nutrients and metabolites. And that is really a very cutting edge area of scientific research, which we are beginning to appreciate and understand better and better, seemingly with each passing month. But functional medicine has a lot to say about that. This term dysbiosis has been used quite a lot too. I think probably most people are somewhat familiar with it, but the idea here is that it refers to an abnormal amount, specific type, or even particular location of these microorganisms within the gut. By and large, dysbiosis is characterized by a state of inflammation within the gut tube. And that can lead to all kinds of issues and challenges, which we will see here shortly. You can see a couple of graphs here. The idea that, as we come into the beginning of the stomach, small intestine, large intestine complex, there's really not that many bacterial numbers. It's really about 1,000 cells per gram of material. And, that makes a ton of sense if you think about it. The stomach has a lot of very strong acid that can be very difficult for bacteria, viruses, fungus to survive and be able to thrive in. Now, there are a couple of exceptions to that, of course, H. pylori, which can lead to the development of stomach ulcers. So some have definitely adapted and are able to work with that. As we move along, basically the bulk of the acidity that is secreted in the stomach as we move into the small intestine starts to become neutralized. The pH goes up, not quite to neutral, but certainly better and more hospitable. And we see that the number of microbiota actually goes up to about 10,000 to 10 million cells per gram. As we work our way into the beginning of the large intestine, 10 to the 10 to 10 to the 11 cells per gram, 10 to the 11 to 10 to the 12 in the transverse colon, and greater than 10 to the 12. That's over 1 trillion cells per gram. That's a ton. We also see that the actual makeup of these different species varies quite greatly by particular location within the alimentary canal, within the gut. And that makes a ton of sense when we consider the pH factor, but also the purpose of these different strains, their potential for competition, their potential for challenge. We're going to talk about all of these factors as well. But this is something that is very important, and this truly is a symbiotic relationship between us, the individual human host, and these microbiota, the symbionts. Um, we support them, they support us when things are working well. Now, what brings about dysbiosis? All kinds of things. Food sensitivities, which is different from food allergies. Food allergy would be what we would think of as an IgG, IgM. I'm sorry, food allergy would be an IgE mediated immune system reaction. Food sensitivity would be the IgG, IgM. So they're different but similar, and they both can result in dysbiosis. And when we have dysbiosis, we necessarily have inflammation within that gut tube. Now, how bad? That's really going to be dependent upon how bad the dysbiosis, what else might be going on. But that is part of the issue that we are dealing with, and one of the big reasons why we would be interested in running one of these comprehensive stool analyses. The idea here with the development of dysbiosis, with the development of inflammation, is that we change the normal state of the mucosal integrity. Ideally, when things are working well, we have these very strong, tight junctions, as they are called. It's basically the connection between one cell in this mucosal lining and its neighbor cell. And ideally, we'd like that to be structurally sound and intact. There are opportunities for these junctions to selectively come apart as a way of absorbing nutrients, but we want that to be done intelligently and as it was intended to be done. When we have the development of inflammation and dysbiosis, we can see the development of intestinal hyperpermeability here on the right. This is what is commonly referred to as leaky gut. And you see that it's depicted here in this picture by these weaker tight junctions and actual full-blown separation of these epithelial cells that line the gut tube. When that happens, our interface between the outside world and our protected inner world is no longer structurally sound. And we have a spilling over of the outside world into our protected inner world. It's like we have just a big rash or ulcer on our arm, and we're just walking around and we're not doing anything about it. We're not bandaging it up. We're not giving it a chance to heal. It is basically direct entry into that protected inner space, which ultimately leads to significant systemic inflammation. This is really the idea of a lot of how functional medicine is structured to practice. I say, you know, it's not all of it, but it's much of it in a nutshell. And I often use this analogy of an hourglass to describe this concept to my patients. At the top, there are all of the potential causes of inflammation, of which there are many. I filled a whole page of PowerPoint with just what I could think of, but there are others. Ultimately, what this leads to at the narrow portion of the hourglass is a breakdown of that intestinal barrier and all of the systemic inflammation that can result from that breakdown. And that spills over into the bottom chamber into systemic consequences. Most directly, most typically, we see this in terms of the central nervous system and the immune system, but it literally can affect every organ system within the whole body. Now, I'm not just saying all of this because this is what I was taught and this is what I believe and this is what I feel. There's really quite a lot of scientific research to back up this whole understanding of how important the integrity of the mucosal lining of the gut truly is. It's been looked at from a number of different ways. This particular paper from 2015 looked at a state of reduced bacterial diversity and found that it was consistent with inflammatory bowel disease. So that would be things like ulcerative colitis and Crohn's disease. And therapies that were undertaken to correct this dysbiosis, which can be attributable to a lack of diversity of bacterial species, showed a great deal of promise in treating IBD. So that's typically done as a fecal microbiota transplant, but we can think at sort of a reduced level, taking probiotics, taking prebiotics, things to support the overall makeup of the microbiome and its robust and varied representation of different species. This paper from 2014 looked at disruption of the gut microbiota, which was how they termed it, and I just wanted to clarify by saying dysbiosis, and how it was implicated in all kinds of various diseases: obesity, IBD, IBS, type 2 diabetes, colorectal cancer. These are inflammatory states like we were talking about at the start of the webinar with respect to COVID-19. Disruption of the gut microbiota, or dysbiosis, definitely plays a role in the development of these types of chronic inflammatory disease states. That breakdown of the barrier, so leaky gut or hyperpermeability, is involved in IBD and autoimmune disease, in addition to the systemic infection. This is from 2018. And then in 2008, a little bit earlier, leaky gut plays a role in the inflammatory pathology of depression. This speaks to that very direct connection between the gut and the central nervous system. And then a very interesting study from just last year took a look at two different populations of very long living individuals, one in China, one in Italy, and specifically got curious about the makeup of these groups of long-lived individuals' microbiomes. And they compared those microbiomes in aggregate to a younger cohort of individuals and basically saw, by and large, that there was greater diversity, both in terms of the individual genus. So that's not quite to the species level, but one level above in the overall biology taxonomy classification of life, as well as at the phylum level, which is really, once you're talking about bacteria instead of animals, phylum is the next degree of differentiation in terms of that taxonomy. So overall, diversity is highly related to living a long and healthy life, it would seem. In terms of practicing medicine, I think most of us are probably familiar with the idea of going to the doctor, getting an annual physical, and having part of that entail some routine screening lab work, whether that's a CBC, a CMP, looking at your thyroid, possibly cholesterol, maybe doing a urinalysis just to make sure that you're not spilling sugar or have a urinary tract infection. Those are, I think, pretty standard and appreciable by most individuals who see the doctor regularly, let's say. From a functional medicine perspective, we have a similar sort of thing. I mean, we definitely appreciate those conventional screening labs, but we also have our own set of more specialized labs. Often enough, these are panels of actual lab tests. And you can see I put the CSA, the Comprehensive Stool Analysis, right at the top of that. That is a really important screen, to my mind, as a functional medicine physician, for the important information that it provides related to this idea of breakdown of the intestinal mucosa and what that can do in terms of all the downstream consequences. Ideally, when we are practicing medicine well, and we have patients who are interested in this sort of undertaking, we're looking to find problems before they emerge as robust clinical pathology. Ideally, we'd like to catch stuff before it emerges as being problematic so that we can course correct and prevent it from ever even manifesting in the first place. So that's one way in which I really like using this test as sort of a preventative screen. But it certainly also does have its place when there are overt symptoms and the situation has gotten a little bit maybe problematic and the person doesn't exactly know why. This can be a way to gain some insight into that. Now, there are lots of other tests that function in a similar sort of way, and I make use of many of these tests in my practice, and we will probably look at some of these as well in future webinars. There are multiple different providers of comprehensive stool analyses. My favorite, the one that I tend to use in practice, is from Genova Diagnostics. Again, I don't have any financial relationship with them. I just really value the completeness of the panel of testing, as well as the layout and the ease of understanding the information that's being conveyed. Typically, there is an out-of-pocket expense from undertaking these tests. Depending upon insurance, that may be about 50% of the total cost of the test. Or if the insurance is not, the most platinum-plated plan available, perhaps. But even not, if it's just very minimalist, they may not contribute as much. Typically, I see prices up to about $400 at most on these tests. But again, if insurance is cooperating, we can see half that or even a little bit less than half that. So this test is often referred to as a comprehensive stool analysis, and I really wanted to highlight that point, the comprehensive part, because we are getting information about a lot of different factors of how the digestive system is working. Digestion, sort of breakdown of key groups of bacteria, what these bacteria are doing for us, the host, whether or not there's dysbiosis, whether or not there's infection of bacterial, fungal, or parasitic origin, and ultimately what the levels of inflammation are looking like. And this, again, referring back to that idea of the hourglass, really gets to this idea of the top half of the hourglass. Digestive insufficiency, dysbiosis, metabolites that aren't sufficient, infection, inflammation, they all contribute to the breakdown of the intestinal barrier. And it's really, every individual is going to be a unique expression of this. It's not just a binary situation. It's really a whole spectrum. How is the overall burden of challenge manifesting in that particular individual? That will speak. And certainly genetics plays a role in this, dietary choices, of course. But this test in particular will give us insight into several of these factors that contribute to that upper half of the hourglass. Now, these are all screen grabs from Genova's test report. This is just a sample subject. This is no individual or patient of mine, so there's no HIPAA violation here. But I just wanted to give everybody viewing this webinar a look at what this report looks like, and you can see how it's laid out. This is essentially the executive summary, first page, which really does a good job of highlighting the key areas. They make effective use of this color sort of scheme. Green meaning, everything is looking pretty good. Yellow, sort of borderline. Red, yeah, there's some serious issues. They also use a particular scoring, and they will make specific recommendations for support based on what the findings show. This would be part of where I would come in as well. I have seen any number of these reports. This is, again, one of my favorite tests to run. So I have a lot of my own thoughts about the significance of these results and how to work with them so that we can bring improvement to the specific individual and their particular circumstances. In terms of digestive capacity, we get a look at levels of digestive enzymes via the pancreatic elastase and what's showing up ultimately in the stool sample collected in terms of products of protein breakdown and specific types of fat. If these numbers are too high or too low, that gives us some insight into how that individual's digestive processes are functioning and what might be needed to better support those processes. This graph here shows the relative commensal abundance. Commensal meaning these are the symbiotic, I guess in this case it would be phyla, so that sort of very high level grouping of bacteria, and shows us, how does this individual sample stack up to a prototypical healthy individual. This can often give us some insight into what's going on, why there's dysbiosis. Each of these different groups of bacteria has a symbiotic relationship not only to us as the host, but to each other. And they perform vital functions for the health of each other. And overall, we want the whole ecosystem to be functioning well. This is essentially the big highlight of the test. We get a breakdown of these 24 different key species and groups of species differentiated by a particular phylum, and we get to see, is this particular strain way out of balance, either because they're too robust or they're just not present. Certain species in particular, like the one towards the very bottom, the Akkermansia muciniphila, is what we would call a keystone species, and there are all kinds of chronic metabolic type disorders that result when this particular species is not robustly present enough within the microbiome. There are things that we can do to correct that, but ideally we'd like to know that that's what's going on, as opposed to just treating blindly based on suspicion. These additional species and groups of bacteria each have their own significance, which is why they're included in this report. Overall, I think at last count, there's something in excess of 1,000 different species that have been characterized in the human microbiome. That's going to vary from person to person, which ones are present, which ones are robustly present, which ones may not be present, which additional species outside of that norm might be present and therefore causing issues. Now, again, one of the reasons why we mentioned that having a healthy microbiome is important is because they take in their own source of nutrition, which is included in the food that we eat. It's typically what we would think of as soluble fiber, which is fiber that's not, it's a carbohydrate rather, that's not eaten for our own direct benefit. We can think of insoluble fiber as basically being non-digestible. It's essentially a bulking agent to help make the passage of stool easier, more consistent, regular, and so forth, as opposed to soluble fiber, which is essentially the food for these beneficial bacteria. They take soluble fibers in, they use it to support and nurture themselves, and then their metabolic wastes are these metabolites, of which there are thousands. I think it's the last that I've seen. And some of the key ones are what we would call these short chain fatty acids. And these are particularly important, so much so that they are actually highlighted on this comprehensive stool analysis test. They are, in essence, a sort of nutritional support that goes directly to that single cell layer of enterocytes that lines the gut tube and maintains that intact, healthy barrier between the outside world and our protected inner space. So when these short chain fatty acids are off, not robustly present enough, our intestinal barrier, that intestinal mucosa, is at a significant disadvantage and is more prone to developing hypopermeability. Genova also has this sort of breakdown of dysbiosis patterns, which they have algorithmically developed based on different scores, which they show here, the inflammation associated dysbiosis score and the methane dysbiosis score. And depending on where the individual's results come back, that would put the individual in one of these 4 zones, each of which has their own particular significance and relevance in terms of treatment choices. Or, if you're in zone 1, then you're probably looking pretty good. You may not need any particular intervention. Now, we also get some key information into how well particular species grow, and that can be species that we'd want to be there, like Lactobacillus, Bifidobacterium, E. coli. E. coli is, there are a lot of different subtypes of E. coli, some of which are very bad, and we will hear about in the news from time to time, and some spinach or whatever that wasn't washed appropriately. But it's not all bad, and much of it is in fact really quite good and beneficial for us. There are other bacteria that get checked out, and the idea here is, are they non-pathogenic or are they potentially pathogenic? Or at worst, are they outright pathogenic? And that is really characterized here in this cultured medium, where we can see how robustly these individual strains grow and the potential significance for how robustly they grow on the overall microbiome. We also get some insight into yeast, Candida in particular, and that can be a big, big problem, and that can definitely cause dysbiosis, inflammation, and lead to all of these downstream consequences as a result. Lastly, we get good insight into parasites, both via what they call a microscopic ova and parasite analysis. So they're actually looking with a human eye, it may be a computer, but it's basically a direct visual observation of the sample looking for eggs or actual parasites. But they also do it via a chemical analysis as well, this polymerase chain reaction parasitology, for some of the more common single-celled parasites. The overt microscopic evaluation will look for the ones that are actually visible with the naked eye as well. But if you're looking just for the eggs or smaller forms of them, then you definitely need that microscope. And then most importantly, some of these inflammatory markers. So calprotectin, eosinophil protein X, and fecal secretory IgA. These each have their own value, but they are a good and broad screen for inflammation that may have set in within the digestive tract. And that would certainly, if this were my patient, warrant additional follow-up action to prevent significantly worse outcomes from coming to be. So that is essentially my review of the alimentary canal, the gut, dysbiosis, inflammation, and what can come about if that arises. It's again a test that is available as an option to anyone who is a patient of Pioneer Valley osteopathy. Currently, just there's drop shipping that's available. It's a perform at home test collection, goes back to the lab via FedEx, and then I get the results through the internet, and then we can do a remote consultation to go over the results. If you are not currently a PVO patient, you can become a patient. We are doing a sort of two-part patient establishment protocol right now, where we can kind of get the ball rolling with the telemedicine consult for the first 30 minutes. And then once COVID-19 settles down and the governor allows us to have non-essential in-person meetings, we would do the second half, which would entail the physical exam, because I got to listen to your heart and lungs and so forth if I'm going to establish a doctor-patient relationship with you. Now, I'm also doing complimentary consultations with anyone, whether you're a patient or not, to go over different recommendations for supplements. I believe very strongly in the importance of supplements, the value that they can bring to overall health and wellness promotion, and really have come to realize that that is a key support offering that I can provide at this time when we're all needing to stay at home for the most part and practice our physical distancing. These consults, again, are available to anyone, whether you're an existing patient or not. And if you are an existing patient, then you already have access to the online supplement dispensary that I use with my patients that carries only pharmaceutical grade supplements. And you already have access to the 15% discount that I provide to all of my patients on everything that FullScript carries. If you want to undertake one of these complimentary 15-minute consults, I'm happy to do that. I'm happy to give you access to FullScript, and I would also be happy to give you a lifetime 10% discount to everything that's available on FullScript. So that comes with specific recommendations from me, tailored to you and your particular needs and what's going on for you. And of course, I'm somewhat limited in how thoroughly I can vet all of those if you aren't a patient, but once you are a patient, we will have undertaken all of those more thorough intake examination processes. We may have undertaken some of these specialty tests, and those recommendations can be just a little bit more particularly tailored. I've mentioned this before, but the idea here with pharmaceutical grade supplements is that you can be assured that your body is getting what you want to give to your body. Standardized doses that have been manufactured under very rigorous quality control standards, and there are no extraneous additives. A lot of supplements that can be purchased through various other mainstream channels, part of the reason why I believe supplements have been given perhaps somewhat of a bad rap in general awareness is because what is being talked about is not strictly pharmaceutical grade supplements. It's supplements that are manufactured with the cheapest ingredients, with an eye towards essentially the bottom line of the producer, so that they can market them at a competitive price and encourage people to buy them based purely on price. But with supplements, as with food, the higher the quality, the higher the benefit to you, the consumer of that nutritional component, whether it's food or a nutritional supplement. It's also important to consider under what conditions the supplement was stored, shipped, and how frequently that product turnover has been happening. It's also an important factor to know that, if you get a supplement that for whatever reason slipped through the cracks or, doesn't work for you, you react poorly to it, that you have good customer service that you can call up and that will be very happy to work with you to address any of the concerns that may have come up for you around that supplement. So again, we've just heard earlier today that we are going to stay in a stay-at-home order for the 2 weeks following the original deadline. Hopefully that will be the end of it, but hopefully if it is the end of it, it will be because that is scientifically indicated to be the end of it. Ultimately, though, we will get through this, and when that happens, I will be seeing new and existing functional medicine patients in person for the whole visit. I will be seeing new and existing patients for osteopathic treatment, all of which, under the current COVID-19 environment, have basically been postponed just because that requires in-person contact. And anybody who is already established would still be able to do a remote consultation. I have patients who reside out of state, and for them that just makes logistical sense, and that's fine. And I will still continue to offer these complimentary supplement consultations as well. So if you have more time and you'd like to avail yourself of any of these options currently, please be in touch with the office. If you want to just wait and see how things develop, that's fine too. Next week, I am planning to do a webinar talking about the organic acids test, which is perhaps my second favorite test if I had to pick one within the functional medicine world. This is an at-home urine collection test. It's really looking at the byproducts of how your biochemistry is functioning, and it is a very broad screen. It's not just focused on one area in the way that the comprehensive stool analysis is, but again, that has implications for pretty much every other organ system in the body, the comprehensive stool analysis. But the organic acids test will give us screening information as to exposure to potential mold toxins, gives us a different way of characterizing intestinal dysbiosis due to the presence of certain bad actor bacteria or yeast. It will give us insight into how the mitochondria are functioning. These, as we talked about last week with Dr. Wilson, are the basically the power plants of the cells. They produce ATP. the cellular currency of energy. And the organic acid test will also give us some insight into nutritional deficiencies as well, and all through an at-home urine collection sample. So let me just take a look and see if there are any questions or comments, and I'm not seeing any. I'm happy to stay on the line for another minute or two. We're right at about the 45-minute mark, the 1:15 time. If there are any burning questions seeking to be addressed, and if not, you are certainly always welcome to get in touch through whatever channel makes the most sense. If you're a patient, you can use the portal. Otherwise, you can email or you can call. There are our office hours as well when we're here. And just want to say thank you one more time for joining in. Really appreciate your kind attention. And just to say one last time, if there's anything I can do to be helpful to you, please be in touch. Otherwise, stay very safe and very healthy out there, and have a great day. Thank you. Bye-bye.